Hello, everyone. Thank you for the organizer to inviting me to here to be with you. It's great to be with you guys, and I think that would be great to take a selfie with you guys. Please smile. Okay. Nice. So today I'm going to talk about the cannabis and what we do at Davoud University on the cannabis genetics and genomics and breeding and all other stuff. But first of all, quick question who I am. It's been years. I'm trying to see who I am. Not successful, but professionally. I did the undergrad in plant sciences. I've worked several years on different labs and even in the level 3 labs, then get tired of the bench work, lab works, and then got to the computational biology, starting working on different types of the genomics project, and got my first license in 2019 when I was at University of Guelph. Then moved to Laval University to start a new program on cannabis, and officially I was the first cannabis prof in Canada, mostly working on cannabis. Université Laval, or Laval University, is the second oldest university in America. It's founded in 1663. It's the largest French-speaking university in America, located in a beautiful Quebec City. So we are mostly French-speaking university. What I'm going to present here is the work of all these beautiful people, all the grad students, postdocs, greenhouse techs. They've done a wonderful job, genomics platform and the metabolomics platform. I'm just here talking and presenting all these results. In my theme, we are focused mostly on 4 different activities in research, and all of them are on cannabis. Mostly we work on the omics technologies like the genomics, metabolomics, transcriptomics. We're developing lots of different tools, technologies to study different phenomena using these technologies. We have an active breeding program. Actually, we are the first public cannabis breeding program in Canada, when we say in Canada, in the world. I have the largest infrastructure to work on cannabis. We can have up to 6,000 plants, so it's huge, so we do that. We have a fully equipped and running genome editing lab. We had a wonderful talk by Mark explaining all the bases of the CRISPR and how it works. One of the questions was how many genes you can now cut. We were successfully to put in 8 genes in one construct and do the CRISPR on cannabis, and it's up and running and working progress. And we have huge activities on big data, machine learning, AI, image processing, and all phenotyping in my group. It's been done based on the image analysis, 3D imaging, and the treatment of the images to understand the different traits. So, why cannabis? In Canada, cannabis now is becoming one of the top economically important crops. Since legalization to 2022, it has contributed more than $43.5 billion to the Canadian GDP. This is equivalent to 25% of all agricultural commodities in Canada, and it has created more than 151,000 jobs. Agriculture is not creating jobs, but cannabis is doing wonderful in that part. It has a great social impact. In 2023, 27% of the Canadians consumed at least once the cannabis. 78% of this population claimed that they've consumed cannabis for medical purposes. And 85% of these people, they've consume the whole plant material. So it shows at what level cannabis is playing a great role in our society. But cannabis is a major crop with a minor research for most of last century, it was banned. We have done nothing on cannabis. It has been left completely in dark. We haven't used any of the, the technologies, innovation, training specifically. I do lots of training or the presentation in different colleges of the College of Physicians, pharmacists, nurses. And unfortunately, in Canada, for example, the physicians, they get trained only 3 hours over 7 years of their training on cannabis. Nurses, 0. Pharmacists, 0. And this is bad. And even the biologists or the students in agriculture, that we have a very big problem in training the people for working on cannabis. So, in Canada, it's been legal, but we are still in the dark. After 5 years of legalization, for example, in Plant Gene Resources of Canada, where we are keeping 115,000 plant species from all around the world, there is 0 cannabis in that gene bank. There is nothing, and even you can't contact these people on talking of cannabis. There is no public funding. The ministries, like the Ministry of the Agriculture, they don't even recognize cannabis as a plant in their chart. So there's lots of problem there. However, they have money to spend on alcohol. So Canadian government in 2022, they've directly invested $166 million in the wineries. So there's lots of problems there, but we are working on that. To be on the same page, because this is a very common problem when I'm talking with our colleagues, physicians or nurses, they are getting mixed up between cannabis and THC. In most of the people, they think cannabis THC, and there is one thing, but they are not really understanding that the cannabis is plants. We can't categorize based on the product of the cannabis, the cannabis. And to arrive to the effect, there is a long and complex process to produce the plants, to do the post-harvest, extract, arrive to the final product, and then the later question is who and what they are consuming the cannabis. That's a big problem in most of the clinical trials when we were talking on the medical side of the cannabis. All people, they are saying we are studying cannabis, but the first question is what cannabis you are studying or which cannabis you are studying. And that's, again, a big problem. That's the thing when we are talking of the medicinal cannabis. That's a huge difference between the modern medicines and the cannabis. When we are talking of the modern drug like Tylenol, that all of you knows that it's composed of 3 main ingredients with a very specific dosage. But when we are talking of the cannabis, we're talking of more than 500 different bioactive metabolites. That is a nightmare for the doctors because they love to have one molecule, one receptor to study everything. But when you're talking that we have more than 500 molecules, they get scared and they don't know how to deal with it. And even for the colleagues who are working on the addiction or other negative impact of the cannabis, this is, again, is complex because compared to other substances that they are mostly one molecules or the specific molecules with a uniform composition, in cannabis we are talking each plant has a different composition. So anyone consuming a different genotypes or strain, they're getting a different effect or different in terms of the physiological impact. So I go back to the biology of high school, how the phenotypes is created. Phenotype is the result of the genetics and interaction with the environment, right? So in cannabis, we are doing a wonderful job in indoor production. We are trying to fully control the conditions to avoid or minimize the impact of the environment. And for the controlling the impact of the genetics, we are mostly relying on clonal propagation. So as I said, for the environment, we have the very nice controlled environment. We can control temperature, water, nutrition, heat, CO2, everything that you can imagine in a very sophisticated way. So we are minimizing the impact of environment. So for the propagation, in general, we have 3 different ways to produce cannabis. First is the getting cuttings from the mother plant, or producing the clones through in vitro tissue culture, or propagating through seeds. So mother plant. In cannabis, you may know we are dioecious plants. We have male and female plants, and we are a very sexist industry because we are only interested in female because the only plants that they produce the high levels of the cannabinoids. And we always try to eliminate the males. That's why we take the female plants with the interesting traits, take them as a mother plant, and try to take the cuttings. So in my lab, we do a great job of having the mother plants taking cuttings, wonderful cuttings of having the nice rooting and everything. Like other industrial partners, we're seeing that after few generations, the cuttings, they're getting deteriorated and they are changing the phenotype, and they are not really similar to their original mother. And that was a question that we were asking, what's happening here. So to study that, we've tried to use the DNA sequencing technology to see what's happening within a plant. So we had a very nice mother plant, 1.5-year-old, taken care of wonderfully well. Everything that was, great to keep it in a vegetative stage for that long time, and it's been that great. So we have taken the samples from different parts of the plants, and we have sequenced the DNA, the whole genome. So how it works, so we extract the DNA, we generate the reads, sequence, and try to align these reads against a reference genome to find if there is differences in terms of the DNA within these samples. This work has been done with my PhD student, Genomic. I think you may know him now. He's the host in the Future Cannabis show. He's doing a great job in that part. When first we got the results, it was just like crazy. It can't be true. So we have redo the whole analysis. We did again sequencing because we found tons of somatic mutation within the plant, within a single mother plant. So that was crazy, because if you take the cuttings from different parts of the plants, you're going to get different plants, different products, different effect. So this was saying that the clones, that is in theory, they should be similar to each other. They are not really clones. They are not uniform, they are not genetically stable, and they have a variable metabolic profile. So at the same time, we have a tissue culture lab. We had lots of clones in our tissue culture system. We've decided to do the same type of analysis for the clones derived from the tissue culture. So we had 78 clones from the same genetics derived from 2 sister lines, that they had the same chronological age, but they were in different number of the generation subculture, from 6 to 11 subculture. And as always, as other industries, we were using SSR markers always to confirm our clonal lines. And when we were doing the SSR markers, very limited number of the markers, 14, that usually most of the collaborators also, they're using all the plants, they were similar, they were like clones, no differences. And this created the question for us, maybe this is a technological problem. So we've taken the same DNA and we did the sequencing by genotyping, meaning we sequenced around 5% of the genome and looked over the variation, and it has changed everything. So there was tons of variation within the clonal lines, lots of, variation within these clonal lines. Another thing that was extremely interesting for us, that was the accumulation of this somatic mutation over different subculture or generation. From 6 to 10 or even generation, we've got the increase in terms of the number of the somatic mutation that has accumulated. This means, again, the clones, they are not uniform, they are not genetically stable, and they have a variable metabolic profile. So, two first methods to produce the cannabis, not very interesting for the medical purposes that we want stability. So we said we'll start doing some work from the seed, propagation from the seeds. The first obstacle that we had, there is no genetically stable cultivars of the cannabis, because mostly we were based on the colonal propagation and the lack of inbred lines. At least whatever we have tested in Canada, there is no inbred line. We are interested in female plants, and then the question comes, do we use the regular females or feminized seeds, and all these stories. So feminized seeds is a very interesting concept that we have in cannabis. Again, we have the male and female plants, the similar chromosomal composition like human. We have XX chromosome for the female and XY chromosome for the male. But we have a luxury and very interesting concept in cannabis, that we can use different chemical or environmental impact. Like here, we are talking of the STS, silver disulfate, that is the ethylene inhibitor. So when we are applying the STS on female plants, it has a masculinization effect. That means the female plant that can produce the male flowers. Then we can auto-pollinate these plants to produce the seeds that they are 100% female or XX chromosome. The same way we can use ethaphone, a chemical that can revert to the ethylene to feminize the male plants and produce the female flowers on the male plants to produce the masculinized seeds. That was very interesting, but we wanted to know how ethylene affects this whole process. A PhD student in the lab tried to figure out all the ethylene pathway signaling and the biosynthesis pathways of the ethylene and all the genes located on different parts of the genome, and how these genes affect the sexuality in cannabis. So, as I said, we have the plants that they are similar either in organs, like the chromosomal composition, or they are similar in terms of the floral organ. So we have tried to divide the ethylene-related genes on 3 different categories. Karyotype concordance, that they are expressed in the same way as the chromosomes. They are based on that. Or they are related to floral organ component, or they are the unique ERGs, to find out the genes that they are involved in sex plasticity. That was really interesting work, and the follow-up works, they are underway for this study. So we are trying to understand the basis of the feminized seeds, because it will be the base of our breeding program. So now we know we need to have seeds. We have to develop a breeding program for the cannabis. So first step to create a breeding program was to create a funding germplasm. So we've done a wonderful job trying to look out everywhere, discussing with mostly every single cannabis industry in Canada to create a representative panel of 200 cannabis extraction representative of Canadian legal market. We have phenotype all these different genetics for more than 20 traits. Even we have created a different descriptor how to study the different traits or the agronomic traits or morphological traits in cannabis. We have genotypes, sequence all these lines with the high-density GBS having more than 800,000 genetic variants, SNP, single nucleotide variants, and we have chemotyped them for cannabinoids and terpenes. During phenotyping, we had some interesting results because for some of these genetics, all of them, they were not coming from the same source. So for some of the genetics, we were able to only have cuttings. For some of them, we had only the feminized seeds, some regular seeds, and so on. During phenotyping, we found something interesting. For example, between the plants derived from a regular female or feminized seeds, we didn't saw that much difference in terms of the phenotypes. But for the plants that deriving from the cuttings or seeds, there was a huge difference. So we had two different categories of the plants, either derived from cuttings or seeds. So the second step was to develop the trait-specific markers to be able to quickly advance the breeding program. So we have done a wonderful job of phenotyping, then try to correlate all this phenotype with genetic variants to find the genomic regions that controlling these different traits. We were able to develop many markers that now we are using in our breeding program to advance our lines. But one thing that was interesting during this analysis of the genetic diversity or population genetic analysis within Canadian legal market, we found that we have a very, very, very low genetic diversity. Everyone's talking that there is lots of diversity in cannabis. There is none. In North America, soybean is famous of not having any genetic diversity. Whole soybean in North America derived from 19 founder lines. The genetic diversity in cannabis we found is lower than soybean. This is scary for the future of the cannabis. And there is no population structure. So we assume that everyone was only interested in THC and forgot the whole concept of the genetic diversity and other traits. Then we optimized the selection practices in our lab. So we have now fully optimized a speed breeding program. So we can go from seed to seed in around 13 weeks. That's great. So we can advance at least 3 to 4 generations per year. That increase our chance of producing new cultivars very quickly. Also, we have optimized genomic selection process for whom are not familiar with that. We have produced a training population, that 200 accession that I've told you. We've phenotype, genotype, chemotype them very extensively. With this data, we have created a model for the selection. Then we do the crosses from the breeding population. As soon as the plants germinates, we take a small sample of the DNA, we sequence, and feed the genotyping data into model to try to predict the phenotype. So we don't wait long to see that. And this helps us to create a very large breeding population. So we produce more than 30,000 plants per year, so we can't phenotype all of them. So based on this methodology, we can do a quick selection process. In terms of the genomic selection, so we had tons of the markers. We've done the single traits genomic selection. Here I'm just showing you the delta 9 THC percentage based on the genomic selection, different traits. So we can, for example, with the random forest machine learning-based tool, we can predict with 60% accuracy the progeny. That is great. That is wonderful in terms of the validation. Then we have developed a new AI-based algorithms that we are now doing a multi-trait genomic selection, integrating traits and also markers. Based on that, we've done, reduce the number of the markers. Now with only phenotyping THCA and 6 markers, we can predict the phenotype with 85% accuracy. That is absolutely wonderful for the breeding purposes, how well we can predict and select the next progenies. So, cannabis research is in its infancy. Cannabis is a plant, and we need to stabilize its genetics. We need to understand the plants before going to do any preclinical or clinical trials. A uniform and reproducible product is required for the clinical trials. That's a huge problem currently, even now we are seeing in Canada, because for the clinical trials, we're getting samples from the stores, which are not the uniform, and creates all these paradoxical results that we are seeing in the literature. Pure lines are the future of this industry. Genomic selection and the speed breeding will speed up this process. And we still need to explore the genetic basis of the different traits and understand how the biology of the cannabis works. Thank you, and I would be happy to take any question. Yes.
Very interesting work. I'm very curious how there seems to be a bit of a contradiction that if you're seeing tissue-to-tissue variation in cannabis, how can cannabis then not be a diverse species? Right? In our experience, when we sequence clones, we don't see hundreds of thousands of variants between clones, and maybe it's just a selection bias of the stuff of the things that we're working with. There certainly could be cultivars out there that have a higher clonal variation rate that we haven't surveyed, but I'm just trying to resolve those two things. If you're seeing hundreds of thousands of variants between tissues, but then seeing very little genetic diversity amongst the hundreds of cultivars you surveyed in Canada, those to me seem to be a bit at odds. Can you explain?
That's a very good question, and I think there is two different concepts we are discussing here. Is the variation within the clonal lines and the somatic mutation that we are seeing within the clones. When we are talking of those thousands of the variants, but it's when we compare to accession, it's not that high, but that's very high within the clonal lines. But when we are, that's the thing. I think the genetic diversity that we are seeing or that exists, I think, is mostly based on the somatic mutation that we are seeing, rather than a real genetic diversity. And I think we have a problem of the founder effect in the cannabis, so we've lost the genetic diversity. And the existing genetic diversity that exists derive from this type of the variation that we see. That's why, it's create this, why there is a variation but not genetic diversity. But I think that's the basis of our genetic diversity, not a real genetic diversity derived from the natural lines. Any other questions? So, thank you.